So just as a reminder, you're not going to be able to see Michael again because he is also having technical issues as his platform. So Michael is a member of the Beyond Myrtle Rust program. He started as a postdoc on this program, and now he leads the pathogen dynamics research area. Part of his research into myrtle rust has been to unpack the evidence of sexual reproduction in Austropuccinia psidii. I'm excited that you'll be sharing your latest results with us, Michael. So if you're all good, feel free to get underway.
Thank you, Marge. Yeah, I'm really excited to be able to share some of the preliminary results coming out of this work, investigating evidence for sexual reproduction in New Zealand populations of the myrtle rust pathogen Austropuccinia psidii. So whether fungi reproduce clonally or sexually is quite important. It impacts the potential for change in those populations, or the adaptive potential of those populations. And that's because sexual reproduction involves recombination, and recombination during sexual reproduction increases genotypic diversity of those populations. That's not to say that clonal reproduction also can't be advantageous. So particularly successful genotypes can increase in frequency rapidly and spread and do well by reproducing clonally. And together, basically that says that a mixed mating strategy, so fungi that are able to reproduce both sexually and clonally, have a really powerful strategy that can help them become invasive and do well in the environment. So when we put this project together, there was a lot of uncertainty about the role of sexual reproduction in invasive populations of this pathogen. And there was also some uncertainty about the life cycle, although that's been resolved now. So to start with, I just thought I would review the life cycle and talk about some of the evidence coming out of what the genotypic diversity of invasive populations from around the world looks like, and then we'll get into the work that we did here in New Zealand. I don't seem to be advancing in my slide. I'll try again. Oh, okay. That's a bit slow. Hopefully it doesn't shoot forward several times. So what do we know about the life cycle for Austropuccinia psidii? Well, rusts can have very complicated life cycles involving up to 5 different spore stages. And for Austropuccinia psidii, 3 of those stages were well known for a long time. So uridiniospores, the clonal stage, had been recorded. Teleospores and basidiospores had also been recorded. And there was uncertainty around whether there was an alternate host, so whether basidiospores infected a completely unrelated host to Myrtaceae, and the other spore stages had never been recorded. Rusts, part of the complexity of their life cycles is that they don't have to do all of these stages, right? So they can have fewer stages, and basidiospores could potentially reinfect Myrtaceae. And so several people had looked into this, and one of the issues was that although they could produce telia and basidiospores and cause infection on Myrtaceae again, they couldn't rule out that contamination of the asexual spores was the result, was causing those infections. So a really nice piece of work led by Alistair and colleagues addressed this by filtering the spore suspension. Because basidiospores are much smaller than the other spore types, they're able to filter that spore suspension and be confident that they only had basidiospores, and then they could reinfect Myrtaceae. So they managed to get infection and pustules produced on the same host, which is really good evidence that Austropuccinia psidii can complete its life cycle on Myrtaceae. And in addition to that, this figure from that paper illustrates nicely the effect that recombination during sexual reproduction has on genotypic diversity in populations. So at the top of that figure there, and next to P1, you can see the genotype, which is the same for all individuals in the parental population. There are 5 loci and 2 different alleles at each locus, and in this case, the heterozygous. So at each allele, at each locus, both alleles are present. But there's only one genotype, which is the combination of all of those alleles at the different loci. After a round of recombination, we can see in the offspring that there are now multiple genotypes present. So there's at least 9 different genotypes there, but the allelic diversity hasn't increased. So there's still only 2 alleles at each locus. And it's important to kind of understand what we're talking about in terms of genotypic diversity, sort of to interpret the results from our work that I'm going to present a little bit later on. So based on the results of this, Alistair and colleagues proposed this life cycle for the rust. So we have the clonal or asexual stage, the uridinospores, and most of you are probably familiar with what this looks like. It produces the characteristic yellow powdery spores on the surface of infected plant material. And this is the stage that really drives disease and epidemics, and it's the one that we're familiar with and we see most often in the field. But the fungus can also produce teleospores, and these look a little bit different on the plant. They're more compact, they're less powdery, and they're brown in color. I've gone backwards. I'm going to go forwards. So teleospores can germinate and produce basidia, and that then produces basidiospores. And on the leaf, that looks a little bit like this. So you can see a brown telium, and it has these kind of white threads coming off of it. So that's what that looks like. And it was pretty hard to see without a microscope, though. And then those basidiospores that are produced can reinfect Myrtaceae and kind of start the cycle again and go in either direction. Right. So now that we're we've got good evidence that the myrtle rust or Austropuccinia psidii can complete its life cycle on the same host, on Myrtaceae. Let's have a look at some of the population genetics of invasive populations of the rust from different places around the world. So for context, the pathogen has spread internationally over quite a long period of time. It was first described from guava in Brazil, and it's native to South America, where there are multiple strains or biotypes of the rust, and those are genetic groupings that are distinct from other groups and associated with particular hosts. In its invasive range, you'll see that most of those records are linked or characterized as the pandemic biotype. So this is a distinct genetic grouping, and there's no strong host association for that group. So they're isolated from lots of different hosts around the world. And then there's also another distinct genetic cluster and an invasive population in South Africa, which is also not host specific. So many of the studies investigating genotypic diversity in invasive populations were finding that there was strong evidence that these populations were clonal. They were only really getting a single genotype. And this figure from the publication cited at the bottom of the slide shows the genotypes and how they relate to one another for samples from different places around the world. So in the left there, you can see the genotype for South Africa, and it's distinct from everything else. We've got some genotypes from the native range at the top of the figure, and then clustering down at the bottom are isolates that are part of the pandemic strain. And all of the isolates from Australia, Colombia, and Indonesia are represented by a single genotype. And similar results were found in places like Hawaii and New Caledonia. Interestingly, though, for New Zealand, even though this sampling occurred sort of early on in the incursion, so myrtle rust had only recently been detected in New Zealand, there was quite a diversity of genotypes appearing, and one of the theories here was that sexual reproduction might be occurring in the New Zealand invasive population. Building on that and going a little bit further, some work was done examining more samples from South Africa, and this time evidence for sexual reproduction or diversity in genotypes was found in the South African population. And I just wanted to show this again because New Zealand isolates were again diverse. So there are multiple genotypes present, distinct from the South African isolates. And there's a slightly different analysis here. This is a network, and we can also see that reticulation, so that kind of square type pattern in the network is indicative of recombination occurring in those populations, and that will be relevant to the results from our work that I'm going to show soon. So what do we do here in New Zealand? Well, We set up long-term sort of monitoring transects at 9 locations in the North Island of New Zealand, where there were populations of Lophomertis trees. Most of those populations were Lophomertis ballata, but we also had a couple of populations of hybrids and the other Lophomertis species, Lophomertis obquadata, at one site. Other native Myrtaceae were also present at these locations. The composition varied, and most of the disease that we saw was recorded on Lophomertis. So during the season in which myrtle rust, the disease is kind of active in New Zealand, that's between October and April, we visited these locations and looked for the sexual spore stage. And we got a relatively good, complete kind of data set for this, although there are some gaps, and they were caused by things like cyclones and pandemics and other things that kind of were a bit disruptive over the last couple of years. And then during the first 2 seasons, we also collected isolates for population genetics work. And we collected across all the sites just over 500 isolates. So collecting an isolate, this is what that kind of looks like. We took a leaf with pustules back to the lab and carefully picked off single pustules under the microscope using a pair of fine forceps. It's followed by simple helix DNA extraction. And then what we wanted to do was use genotyping by sequencing analysis, which required high molecular weight DNA of a certain quantity to happen, and it's really hard to get that from an individual pustule. So to make this possible, we performed a whole genome amplification step using QIAGEN's repli-G kit. All of these whole genome amplifications were carried out in a hood. We were very careful to avoid contamination because the whole genome amplification kit is not specific, so it will amplify any DNA that's in the sample. And we, for a subset of samples, when we first kind of got this up and running, we sequenced the ITS region, and we were getting hits back for Austropuccinia psidii. So that was indicating that what we were doing was working, and we were getting DNA for our target. So that was good. We then proceeded to amplify all the samples and send them to our sequencing provider, AGRF, over in Australia. And then they performed the GBS analysis. So that involves restriction enzyme digestion. So the DNA or the genome is broken up into pieces, and then pieces of that genome are sequenced, and you end up with a reduced representation sequencing of the genome. AGRF then complete part of the bioinformatic analysis. So they do the SNP calling part, and they mapped those SNPs to the reference genome for Austropuccinia psidii. And then Alistair used a variety of programs that are listed there to do some filtering on the data set and some analysis, and we'll kind of get into that a little bit later. So what did we find? Let's look at what we found in terms of observations of Telia first. So we found the sexual spore stage at least once during our sampling period in 8 of the 9 populations that we were visiting. The only location where Telia were not recorded was the site in the Kaimai Ranges, sort of in between the Bay of Plenty and the Waikato region. We found Telia fairly consistently from 2 sites in the Taranaki region. And it maybe there's some kind of environmental driver that's sort of pushing that. Another key point I think that's important to get across is that Telia were quite often cryptic. So the bottom right photo there shows a couple of Telia sort of mixed in with several Uridinea. It's pretty hard to see them when you're out in the field when they're like that. It's easier to spot them when they're kind of more abundant on a leaf. So I think it's reasonable to say that we're probably missing some of the Telia that are there. We're not finding all of them when they're present. This data shown in the table is only for Lophomertis, so that is either Lophomertis ballata or obquadata or hybrids, depending on the location. And most of our observations are on Lophomertis ballata, but that's heavily biased by the fact that most of our sites contain that host. We also recorded Telia on Syzygium myrti in 1 instance. And it's worth noting also that herbarium specimens collected early on in the incursion here in New Zealand that include Lophomitis ballata from Taranaki, and Kermadex pahutakawa from Roll Island also had Telia on them, so they were checked. Okay. Now, if we pull all of the data for our observations of Telia from all sites, and we look at whether there's a seasonal pattern, apart from the first year where things are slightly different, not many observations are made and they're over a sort of broader timescale, we get quite a consistent pattern where we're only really seeing Telia between January and March. And this is kind of interesting. It's when the disease activity for Austropuccinia psidii peaks in New Zealand. So it's when we have the highest abundance or incidence of disease on trees at these locations. And it's also when disease is kind of most severe, usually around February, in the middle of that period. So observations of Telia sort of align with incidence and severity of the disease. Okay, so moving on to what we found in terms of population genetics analysis, because observations of the sexual spore stage are great, but they don't really confirm that the sexual reproduction is actually occurring. We need to kind of pair that with what's happening in terms of the genotypic diversity in those populations. So we got just under 14,000 SNPs coming back from all of those, the 522 isolates that we sent to AGRF. And all of those SNP sites were mapped back to the genome assembly for Austropuccinia psidii. So that's good. It confirms that they're not contaminants, that we're actually looking at DNA from our target. We then had to do some filtering because there was quite a bit of missing data in the data set though. So the first thing we did was get rid of any individuals that were missing more than half the data. And that brought us down to 379 isolates. We then filtered SNP sites where they were present in fewer than 250 of those isolates. So the final data set that we ended up with was just under 12,000 SNPs and 379 individuals. And we had on average about 20% missing data. So what we ended up with was still a sizable data set with a lot of information, and we were very happy with that. The first kind of thing that we can take away from looking at that data is that there was a low average nucleotide diversity, and we also had a negative Tajima's D statistic. So this is the result that you'd expect for invasive populations that have low founding diversity and then undergo sort of a rapid expansion. And that's exactly the what happened with Austropuccinia psidii here in New Zealand. We expect a low founding diversity of spores coming from, most likely from Australia, where genotypic diversity is recorded as being low, and then a rapid expansion across parts of New Zealand. So the next thing we did was look at k-means cluster analysis to see if there was any structure in the populations based on either the year in which the samples were collected, the host that the samples were collected on, or the location where they were collected. And there's something we have to fix up here, that the Lophomitis obcordata and the hybrids were lumped together in the data sheet, so we have to kind of split those out, but I don't think that really makes any difference to the overall conclusions. Basically, the clustering analysis revealed no strong structure based on any of those criteria. If they did, you'd expect to see so basically all one color in one block and all one color in another block, and that's not what we see. We see a mixture of things. So if clonal reproduction predominated, we'd expect to see some kind of stronger structuring. Then we looked at the data using a network analysis. There's quite a bit going on here, so we'll talk through it. But this kind of backs up what we saw with the k-means cluster analysis. So there's no strong pattern in the network. You can see that isolates collected from the same locations or the same hosts or in the same year are spread throughout the network, and they don't group together. One of the nice things about this analysis is it also provides another measure of kind of robustness of our data. So in the data set, we included a number of technical replicates, and these should have the same genotype, right? Because it's the same DNA. Obviously, they don't come out exactly on top of each other because of the missing data element, but all of them were recovered right next to each other in the network. And they're indicated by these kind of dotted lines that join them together, and I put a few arrows in there kind of pointing out where some of the markers are a little bit hard to see. Based on the positioning of those technical replicates, we can kind of make a decision about what a clone should look like. And we see some evidence of clonal groups that cluster together near the external parts of the network. But overall, we have a really strong, highly reticulated central area forming a kind of star-like pattern, which is a really clear signature of recombination in a network analysis. And given that we had no evidence of structure in the population, we also calculated the index of association across the entire data set. In clonal populations, the index of association should approach 1, and what we got was 0.01, so very low, and this indicates that there is random inheritance of SNPs, and this also supports that recombination is occurring in these populations. So in summary, our hypothesis, we kind of expected that there would be a limited number of genotypes that were predominantly spread by clonal reproduction, and there might be a little bit of sexual reproduction occurring. But based on the data that we've got back, we reject this. The data set, the population genetics data set, has hallmarks of an invasive population under a founder effect, which is good. That makes sense because that's what we expect to see. There's high genotypic diversity, but low allelic diversity. There's no evidence of linkage amongst loci based on the index of association. There's a complete lack of population structure, and we found sexual spores produced in most of the populations. And all of this evidence together kind of suggests that sexual reproduction is relatively frequent in populations of the rust here in New Zealand. And this has some important implications for management. So it suggests the populations of the rust here will be quite adaptable. And what that might mean for current resistance in endangered host populations is uncertain. Any programs looking to screen for host resistance and breed host resistance need to take that into account. It highlights that we need to take the development of fungicide resistance in Austropuccinia psidii populations really seriously and implement all of those measures to reduce the risk of that happening. And it further highlights the potential risk of outcrossing with a strain that's not currently present in New Zealand occurring if a new strain of the pathogen arrives in New Zealand. So I just thought I'd put up some references that link back to some of the stuff I talked about in the introduction, and people can kind of pause the video and look at it if they need to. And I've got a lot of acknowledgements that I'd like to make for people that have contributed to this work over the last 4 years or so. It's been a big effort, so thank you very much for everyone that's been involved. And that's me finished. So hopefully that was okay. And if anyone has any questions, I'm happy to try and answer them.
Thanks so much, Michael. That's really, really interesting. And I'm happy to start fielding questions from the audience. So audience, if you can just pop them into the chat. I'm going to start with a question. So I was actually really surprised about the amount of diversity that there was present in, like, even in the the initial years of the incursion. Does that mean that there were multiple times that the spores came, flew over from our neighbors?
Well, yeah, potentially. So it could be the result of multiple introductions of different genotypes coming in. The alternative is that we had sort of early sexual reproduction occurring shortly after arrival of the pathogen, which produced that genotypic variation here in New Zealand. So I think either of those could explain that pattern, but it is pretty surprising.
Yeah, so it couldn't be possible that there was just one event. It possibly could have happened multiple times then.
Yes, yeah, entirely possible. Yeah.
So I guess that being said, and I know that you mentioned about the like keeping, well, I mean, even though that there is so much diversity present now, Austropuccinia psidii is an unwanted organism, and do you feel like that this is something that we should carry out into the future, that to make sure that it stays on the unwanted list, even though it, you and I know it definitely makes it difficult to do research on it?
Yeah, it's an interesting question. I think we definitely don't want additional strains coming into New Zealand. So those biosecurity measures to prevent that are really important. I suppose it's the incursion that's already occurred was sort of natural dispersal on the wind, which we couldn't have prevented anyway. So it's an interesting one from that perspective. Yeah, I think that unwanted organism status is quite important, but we will need to sort of monitor what the situation looks like, and I think it's important for us from a New Zealand perspective to be monitoring what's happening in neighbouring countries. So if a new strain or variant arrives in Australia, then that's certainly cause for concern. But I don't know if there's anything that we can really do to prevent it from coming here to New Zealand anyway.
Yeah. So do you have some comments about how the different spore types and reproduction affects overwintering and survival, especially in times of little to no plant growth or longer and cooler winter periods?
Yeah, so I mean, a lot of the research on rusts looking at life cycles was done on rust in temperate regions, lots of it on cereal rust, and often in those cases the sexual stages are kind of overwintering structures, so they have thick cell walls and they can survive during cold periods. Austropuccinia psidii is a tropical species. It doesn't really have thick cell-walled teleospores, so there's not really strong evidence to suggest that it's produced as an overwintering mechanism. It's more likely, I think, here that overwintering occurs inside the host plant, and Rob Beresford's kind of talked about that in his previous webinar as well. I think infection inside the host overwinter, and then maybe very intermittent clonal cycling when it's possible during that time, or just latency throughout winter is more likely to be the overwintering mechanism here.
Cool. So have there been any similar studies in Australia, and do you think that the situation would be similar even with different genera and species of Myrtaceae?
Yeah, those are really good questions. So there was work done in Australia. I mean, and I kind of talked about it in the intro, and they did a wide amount of sampling and found a single genotype, right? So that's quite surprising. That work was done using microsatellite markers, so a different method to what we've used here, and that sort of samples a much smaller part of the genome than a GBS analysis, which has much better coverage. So potentially using some of these more modern techniques, they might find something different is going on in Australia. Sorry, what was the other part of the question, Marge?
Oh, sorry, I'm just.
Oh, different hosts, right? Yeah.
Yeah. Yes, different hosts.
So yeah, that's a really interesting question, and we were hoping that we would be able to address that. We had a master's student in an aligned project, and we collected isolates from Metrosideros diffusa, which was the other species that we found rust symptoms on, nowhere near as often as we found symptoms on Dilophomonas. Unfortunately, the lab part of that, in terms of the whole genome amplification side, didn't work very well, so we couldn't pursue that further. yeah, I mean, given the pattern that's found in the native range of the pathogen, and this kind of tendency to have evolved towards specific genotypes on certain host groups, over time, that certainly could be what happens here, and we'll just have to monitor that situation.
So lots of compliments for your talk. So was there any clonality within the populations at all?
Yes, there was. So there was some evidence of clonality in that network. I don't know if I go back. Marge, can you see my mouse? Is that showing up or not?
Yes, I can.
It is. Okay. So, for example, this little group here is probably a clonal group, and those are all collected from one location around in the same period of time, and then in the following year. So there is evidence of clonal of a mixed strategy, but we found that there was evidence for greater levels of recombination and sexual reproduction than we expected. So that was the surprising part.
Yeah, really interesting. So do these results mean that plant populations will struggle more to resist and survive myrtle rust?
That's a really good question. I mean, the key takeaway message is that the populations of the rust here in New Zealand are expected to be more adaptable than they would be if they were only reproducing clonally. That doesn't necessarily mean that they will evolve towards being more aggressive or more virulent on different hosts. And often we actually, it's not in the best interest of the pathogen to cause a lot of damage on its host. So it'll be interesting to see what happens. But I think it's more important in terms of a management context where we are sort of actively using things like fungicides or keeping a high number of hosts, potentially of low genetic variation in one place, like, for example, in nurseries, or if we're planting trees out. So we have to be really aware of this adaptive potential in the populations here and take that into account when we're looking at putting trees out into the environment or caring for them in sort of modified environments.
So this is a question. Do you think that long-term monitoring could be continued to inform potential host response changes and landscape changes? Like we've definitely struggled with keeping this going, but they seem to be very informative.
Yeah, long-term, I mean, if we can get resourcing to continue sort of long-term monitoring, that would be fantastic. In terms of sampling and looking at the population genetics, it's pretty resource intensive, and it's taken us a long time to get to this point. It wasn't straightforward. So developing tools that enable that kind of amplification of polymorphic sites or SNPs, for example, would be really worthwhile to make it easier to kind of look at the differences or generate genotypes for isolates without having to go through this process of trial and error like we have had to. And I guess our work can sort of help inform that potentially in the future, which is good.
Yep. So were SNPs mapped down to specific trees or leaves in your data sets?
We haven't done that yet. I think in the analysis, I'd have to go and check with Alistair, but I don't think so. So this is kind of preliminary. These are the sort of first main things that we wanted to look at, and we've obviously got the potential to look at quite a few interesting things. So that's a good suggestion, and I'll see what we can do.
Okay, awesome. So I think that since we're running out of time, I'm just going to ask one last question. And do you have a sense of how many introduced Myrtaceae species that myrtle rust has been detected on?
off the top of my head. There is a list on the myrtlerust.org website of all of the hosts recorded in New Zealand, so including native and exotic species. So I don't recall the exact number off the top of my head, but it is available online.
Yep.
I don't know if you know much.
Oh, I was going to say, I was like, nope, I think that that's probably a good answer, that myrtlerust.org does have that information. And as always, if you happen to see myrtle rust on something that hasn't, on a host that hasn't been reported before, then be sure to put it on iNaturalist, because that's good.
Yeah.
So I think that that's all that we have time for today. Thanks, Michael, for your presentation. And so a video of today's webinar is going to be made available on the Beyond Myrtle Rust website in the next couple of days, and it'll be emailed to everyone who registered. Our next webinar is on June 12th at 11 a.m. New Zealand time, and that is going to be presented by me. And that's because the Beyond Myrtle Rust program is ending June 30th. So I'll be talking about how far we have come and what's going on for the road ahead. And you'll have to just stay tuned to see what's going to happen after that. So with that, keep the lookout for the webinar invite for next month. Thanks, everyone. Hi, Rara. See you next time.